Welcome. This is our fourth Organization of Biological Field Stations, OBFS, live from the field event. My name is Carrie Winninger with Sonoma State University's Center for Environmental Inquiry. Keep an eye out for details of 4 more events that will take place next spring, including topics such as remote sensing of biodiversity, effects of herbivores on landscapes, carbon storage and cycling, and science and industry responding to climate change. Very quickly, I just want to go over some guidelines for this webinar. Remember, all participants are muted and your video cameras are off. During the presentation, please use the chat button to communicate with the panelists and attendees. Remember to choose all panelists and attendees so that everyone will see your comments. During the presentations and the live Q&A session with researchers, please submit your questions using the Q&A button, not via the chat window. If you're here as part of a college class, please type the word student in front of your question so we can prioritize your questions. And it's important to make sure your full name is visible, either as your Zoom username or typed in after the word student. This is so you receive credit for attending today. Also, welcome if you are watching us streaming live on Facebook. We will try to get to your questions too. Without further ado, I will turn this over to Elizabeth Borer, Professor of Ecology, Evolution, and Behavior at University of Minnesota. She is a researcher who studies the effects of global change on disease, biodiversity, and nutrient cycling in grasslands.
Thank you all for joining live from the field. We welcome classes, faculty, and other participants from across the nation and around the world. Welcome, everybody. This is the 4th Live from the Field event. In this series, we take you on virtual field trips to research sites around the country and the world. As you'll see today, we're visiting 3 biological field stations, places where people from many different backgrounds come together to study the environment. Live from the Field events are a project of the Virtual Field, an international coalition of over 50 field stations and marine laboratories. This National Science Foundation funded effort brings the field to you. Find out more about events like this and other virtual learning experiences for college students at thevirtualfield.org. Today, we're traveling to field stations in Argentina, Colorado, and New Mexico to talk with researchers studying the impacts and feedbacks of nutrients like nitrogen and phosphorus in the world's grasslands. I've been studying the effects of global change on disease, biodiversity, and nutrient cycling in grasslands for 20 years. I love field research because I get paid to do what I love, spend time outside being active, staying curious, observing, and collecting data on the natural world. Part of my interest in grasslands is because they cover about 40% of Earth's land surface. Agriculture and fossil fuel combustion are adding huge amounts of nitrogen and other elements into these natural environments. And agriculture is also changing the types of herbivores in these landscapes. But we're only beginning to understand how these changes affect the functioning of grassland ecosystems. The researchers you'll meet today are working to tease apart the effects of climate change, nutrients, and herbivores to understand what future Earth will look like. All of us are working as part of a collaboration of scientists in 27 countries called the Nutrient Network to understand nutrient cycling in the world's grasslands. Each researcher has prepared an 8-minute video describing their research into the impacts and feedbacks of nutrients in grasslands in a changing world. After the videos, the researchers will be available to answer your questions live. I'd like to encourage everyone to post questions in the Q&A as we go along. Students, remember to type student and your full name in front of your question. So we'll start at the Central Plains Experimental Range, which is a research station owned by the US Department of Agriculture. This site is located at the western edge of the Pawnee National Grasslands in northern Colorado. A lot of research at the site seeks to better understand relationships among climate, vegetation, and livestock production. The site has been home to ecological research networks, including the International Biosphere Research Program, the National Ecological Observatory Network, and the Long-Term Ecological Research Network. And now it hosts one of the Nutrient Network experimental sites. The researcher you'll hear from first is Dr. Cindy Brown, who's a professor at Colorado State University in the Department of Agricultural Biology, where she teaches and advises research projects for undergraduate and graduate students. She's especially interested in the factors maintaining plant species diversity and why new species can invade and sometimes dominate grasslands. She's been working at the Central Plains Experimental Research Station for 10 years, and one of her favorite parts about field research is that it gives her an opportunity to spend time outside getting to know the plants and animals in an ecosystem.
Hi, I'm Cindy Brown. I'm a professor at Colorado State University, and I have a master's in biology and a doctorate in ecology. So my specialty is plant community ecology, which is the study of plants growing together in the same area and how they interact with each other. I also am especially interested in invasive plants and weeds and the study of restoration of disturbed systems. So I like to work at the interface between restoration ecology and invasive plant management. I'm passionate about doing research that tests basic ecological theory at the same time as it answers questions that are important for science-based management. I grew up in a rural area, and I spent a lot of time in the grasslands and creeks around my home. And I was very interested in biology all along. So I wanted to be a medical doctor when I was in secondary school and also at college. After I graduated, I signed up for the Peace Corps, and that was a transformative experience for me. So it made me ask how I might be able to use my my science background in the conservation effort. So, until I found ecology, I just had no idea that I could get a job studying the plants and animals that I loved so much hanging out with when I was a kid. I'm talking with you today from the Central Plains Experimental Range, which is a U.S. Department of Agricultural Research Service field station. It's located in northeastern Colorado, which is more or less in the middle of the country. And it's on the plains on the eastern side of the Rocky Mountains. It is around 5,000 feet in elevation, and it's part of the shortgrass steppe or shortgrass prairie ecosystem. The site is within the historic range of bison, but they were eliminated about 150 years ago. There were and still are pronghorn antelope, mule deer, jackrabbits, prairie dogs, and other small mammals at the research station. Cattle were introduced along with settlers and still graze the research station as part of research on their effects and also as part of the management of the rangelands. So the plant community here is the shortgrass steppe, and it's dominated by short-statured grasses, in particular blue grama and buffalo grass, which are warm season grasses, so they reach their peak biomass late in the summer. There are also cool season species here of sedges and grasses, and although they're not as abundant, there are a lot of species of forbs as well. So these are herbaceous broadleaf plants. Some of them have beautiful flowers, and their abundance depends a lot on the weather in a given year. So in this system, about 80% of the biomass is below ground. So we're conducting an experiment that's part of the nutrient network, and it includes treatments that, one, exclude large hooved grazers like the pronghorn, and animals down to about the size of rabbits. Smaller than that, the animals can probably get in. And a second treatment, set of treatments is nutrient addition. So we use these two factors because changing global nutrient budgets and changes in the identity and abundance of different herbivores are two of the biggest effects that humans are having on ecosystems globally. So the global pools of nitrogen have doubled, and global pools of phosphorus have quintupled since, and this is due to the burning of fossil fuels and also agricultural fertilization. Some herbivores have been removed from food webs through hunting and habitat loss, and others have been introduced either intentionally or unintentionally by humans. So the nutrient network was established to study the effects of these human-caused changes in grasslands across the globe. So the main questions we have are, what are the global patterns of relationships between productivity and species diversity? And how are plant production and diversity affected by limitations of multiple nutrients? And how do changes in grazing and fertilization affect plant biomass, species diversity, and species composition? So I love being able to work outside in beautiful places like the shortgrass steppe while answering interesting scientific questions that can help us better manage rangelands and natural areas. To study these questions, we set up the standard nutrient network experiment, which has 10 treatments replicated 3 times. Each replication includes all 10 treatments grouped together, and this is called a block. So the nutrient treatments include nitrogen, phosphorus, and potassium plus micronutrients. And they're applied to different plots alone or in pairwise in pairwise combinations, and also all three together. There's also a control plot that receives no fertilizer. For the plots that are assigned the grazing exclosure treatment, they have fences, and one fenced plot has no fertilizer added, and the other fenced plot has nitrogen, phosphorus, and potassium plus micronutrients added. So the plots are 5 meters by 5 meters, or 25 meters square, and we divide them into 4 quadrants and apply or do different sampling in different quadrants. So one of the quadrants is used for measuring species composition and abundance, and also measuring light availability. So these are non-destructive measures. Another quadrant is assigned to destructive measures, such as in particular, the clipping of biomass. So we also take soil sample measurements in those quadrants as well. Overall, we're finding that the distributed experimental network is a very powerful tool for testing basic ecological theory. We're also finding that species diversity promotes ecosystem stability and productivity. And thirdly, we're finding that nutrient supply and herbivory control species diversity via changes in composition, both by introduction of non-native species and loss of native species. Here at the shortgrass steppe, we're finding that the native dominant warm season species are being replaced in the fertilizer treatments by shorter-lived cool season native species, and also a non-native winter annual. We're also finding years with large biomass production when there's plenty of rain in the fertilized plots, followed by years with very, very low production. And finally, we're also finding that the ungrazed fertilized plots are often dominated by weedy forbs like tumble mustard. So there are many opportunities for students to get involved in the nutrient network research. We can always use help applying nutrients in the spring and collecting biomass data in the late summer. Also, there are opportunities to work with the extensive network-wide database if students want to explore questions, ecology questions that they have interest in.
Fantastic. Well, the University of New Mexico's Seviata Field Station is located in the 220,000 acre, or about 90,000 hectare, Seviata National Wildlife Refuge in central New Mexico. It's about 80 miles from Albuquerque. It was initially established in 1989 to support the research efforts of the Seviata Long-Term Ecological Research Program. Research at this site promotes understanding and preservation of the natural environment with a focus on the effects of a changing climate and biosphere-atmosphere interactions. This location is particularly good for this research focus with the Chihuahuan Desert to the south, the Great Plains to the north, and the shrub steppe to the west. Dr. Scott Collins is a professor at the University of New Mexico. Scott has been working at the Seviata field station for over 17 years, studying the effects of global environmental change on grassland ecosystems. He loves everything about field research, especially being outside, observing dryland ecosystems, and marveling at their resilience. In his research, Scott is especially interested in how heterogeneous environments control plant communities and how plant communities change in response to disturbances like fire.
Hello everyone, and welcome to the Seviata National Wildlife Refuge. This is the home of the University of New Mexico Seviata Field Station and the Seviata Long-Term Ecological Research Program. My name is Scott Collins, and I'm a professor in the biology department at the University of New Mexico, and I study the impacts of global environmental change on grassland ecosystems. The Seviata National Wildlife Refuge is located about 50 miles south of Albuquerque, New Mexico, and it's in the very northern end of the northern Chihuahuan Desert. And we are standing here today in a beautiful and expansive desert grassland. So even though we're in a national wildlife refuge and we're far away from major metropolitan areas, the refuge is still impacted a lot by global environmental change. In particular, there's a lot of atmospheric deposition that we experience here in this desert ecosystem. And so perhaps you can see in some of the panoramas around here the smoke and haze that you find off in the distance. Especially in the wintertime, we have a lot of fires and smoke from the western United States, and a lot of the smoke that you see in the air here is coming from wood-burning stoves in the communities to the north of the refuge. We're experiencing a lot of atmospheric pollution down here, and so this refuge is actually impacted quite a bit by human activities. Now, deserts are very low in nutrients in general, and this air pollution that you see is going to be adding all kinds of nutrients into this ecosystem. And so we're interested in determining what are the consequences of increased nutrient deposition from atmospheric pollution on these desert grassland ecosystems. So what we do in response to this atmospheric pollution is we do experiments by adding nutrients to these ecosystems. And so what we want to do today is introduce you to one of our long-term nutrient addition experiments, which is part of the Nutrient Network, a global experiment looking at the impact of nutrient deposition on grassland ecosystems around the world. So follow me and we'll head out to the experiment. Are you ready? Let's go. So as I said earlier, the Seviata sits at the very northern end of the Chihuahuan Desert, and it's actually in what's called an ecotone, and we are at the dividing line between the Chihuahuan Desert, which extends to the south, and Great Plains grassland, which extends to the north. As a consequence, there's a nice mixture of species from those two ecosystems that occur here together. We would anticipate, under climate change in a warmer and drier environment, that the Chihuahuan desert species would be increasing in this environment, whereas the more mesic and cooler species from the Great Plains grasslands would be declining. And that's what we're interested in finding out here in this ecosystem. And we also want to know what are the other environmental drivers of the dynamics between those two ecosystems, the Chihuahuan Desert and the Great Plains grassland. So one of the big questions is, how do those species respond to nitrogen deposition and other nutrients in the soils? Desert soils are very, very low in nutrients to begin with, and so the atmospheric pollution that we're getting here year after year after year is bringing lots of nutrients into this ecosystem. So the Nutrient Network experiment is essentially a dose-response experiment where we increase the amount of nutrients that are available in this ecosystem and ask questions about how the species and ecosystem processes will respond to this increase in nutrient availability. So one of the things that we've learned through this long-term nutrient addition experiment as part of the Nutrient Network is that one of the dominant grasses, the Chihuahuan desert grass, black grama, does not like nitrogen deposition and added nutrients in the soil. And we see a decline in abundance of the species that should be increasing under a warmer and drier climate. Instead, the species characteristic of the Great Plains grassland responds very positively to nitrogen addition. And so we see that that species is actually doing better under nitrogen fertilization. But one of the strongest responses we're finding in our ecosystem is exhibited by the most common invasive species that we have in our grasslands here, tumbleweed. Tumbleweeds are annual plants. We think of them as nitrogen hogs. And if you look at the experiment in the background here, you can see some large dark plants. And those are the tumbleweeds that have benefited from the nitrogen deposition. And they essentially define the plots in which we have added nitrogen to this ecosystem. The nutrient network doesn't just add nitrogen. Also, the experiments add phosphorus and potassium, and they're added in all possible combinations. Although we add nitrogen, phosphorus, and potassium into this ecosystem, we find that the system does not respond very much to the addition of phosphorus and potassium. It responds primarily to the addition of nitrogen. And what is most important is that dryland ecosystems like this grassland that we're standing in are very, very dependent on water. These are what we refer to as pulse-driven ecosystems. Nearly 2/3 of our annual rainfall occurs during the summer monsoon season when we have rain events that essentially are pulses that turn on these ecosystems and establish and start ecosystem processes. And it is only under those periods of time when there's soil moisture available that we can actually get a nitrogen response in the system. So one of the questions we've asked from looking at the response of this system is what has happened to all the nitrogen? We have added a tremendous amount of nitrogen to this ecosystem year after year after year. And it turns out that when we analyze the soils from these experiments, we find very little accumulated nitrogen in these soils. And this is a surprise to us. And so we have to ask, where is this nitrogen going? And we think that some of it is, of course, used by the plants for increased growth rates, especially the Great Plains grass. But much of the nitrogen that gets added to this ecosystem volatilizes out of these soils back into the atmosphere, because it turns out that these soils are very, very hot in the summertime, which creates this reaction. So we lose a lot of the nitrogen that actually comes into these soils through this fertilizer application. So we started our nutrient network experiment back in 2007. We got involved in the nutrient network because they sent out a message asking people to volunteer to follow their experimental protocols to be part of a globally distributed bottom-up experiment in which all the participants join in this effort following similar protocols so that we can have a better understanding about how global grasslands will be responding to changes in nutrient availability. We felt it was important from the Seviata standpoint to contribute to this network because we are at the drier end of the moisture gradient of grasslands. So as I mentioned earlier, I'm a professor in the biology department at the University of New Mexico, and I love what I do. I get to work outdoors, and I get to study in nature. And I got to do this because when I was an undergrad, I had a professor that took an interest in me and helped me. So what I recommend is if you're interested in this kind of life, talk to your professors. They're always excited to find students that are interested in what they're interested in, and they will work hard to help you meet your career goals.
I love that message. All right. So next, Las Chilcas is a field site located in a 5,000 hectare, about 12,000 acre region of the Pampas grassland to the west of Buenos Aires in Argentina. For those of you unfamiliar with this part of the world, the Pampas grasslands are vast plains that extend through most of the middle of Argentina from the Atlantic coast all the way west to the foothills of the Andes Mountains. The study site is owned by the Institute for Agricultural Plant Physiology and Ecology that aims to conduct basic and applied plant biology research related to agronomic problems and to the sustainable use of natural resources. Much of the research at this site focuses on climate, vegetation, and livestock production. Dr. Laura Chatian is a professor at the University of Buenos Aires and a researcher at the Institute for Agricultural Plant Physiology and Ecology. Laura researches the effects of drought, species invasions, grazing, and nutrients on plant diversity and plant growth, or primary production, in grasslands and deserts. She also studies what controls decomposition of plants, which is important for understanding the uptake and release of carbon dioxide from the atmosphere. She's been conducting research at Las Chilcas for 12 years and enjoys field research because it gives her a better understanding of the natural world that will ultimately help to improve nature's benefits to people.
Hello, my name is Laura Yatejian. I am a professor at the University of Buenos Aires, in Buenos Aires, Argentina. I'm an independent researcher from the CONICET. I am a biologist by training, and I made my PhD in agronomy. I'm actually doing research and teaching at the Faculty of Agronomy at the University of Buenos Aires, Argentina. My discipline is ecology. I am a field ecologist. My main questions are related to the effect of different global change drivers on ecosystem functioning and plant community structure. This is my field site, Las Chilcas. We are in Argentina, South America, in Buenos Aires province, and the area is a big grassland without trees. The vegetation is very low, as you can see, and the landscape is flat. This is a vast area of grassland devoted to livestock production. And we are here interested in understand the effect of different global change drivers like land use, temperature increase, and the higher frequency of extreme events like drought and flooding. And also we want to find out the effect of the increasing nitrogen deposition and increasing phosphorus stocks in our grassland. To do that, we establish manipulative experiments in the grassland, and we compare the plant biomass production and plant diversity in manipulate plots where we change the different factors with control plots that are just natural grasslands devoted to the common conditions. The plant community is composed by different species. The plant species can be arranged in plant functional types, and we can identify here legumes, other forbs, and grasses. We don't have trees because this is a grassland, and the plant species composition change with the addition of nutrients. In this plot, we have added nitrogen, phosphorus, and potassium for several years, and the plant species diversity decreased with nutrient addition, but we increase the plant biomass. Also, with this nutrient addition, the cows are more interested in this plot comparing with the background. So the height of the plants are smaller because this plot is more grazed than the background grassland. To estimate plant cover, we use this photosynthetically active radiation device, which is a 1 meter long bar that we deploy at the ground level, and we compare the amount of incoming photosynthetically active radiation inside our experimental plot where we have been adding nutrients with the background grassland outside the plot. That you can see here, it's has more plant cover, so less incoming radiation is received in this plot. To study the impacts of grazing, we exclude cows from our plot with this exclosure, and we compare the same measures, for example, the plant species composition and above ground plant biomass inside and outside the plots. As you can see, there is a big difference in the plant biomass in both cases. To study the effect of drought, we deployed this rainout shelter, which consists of bands of acrylic that are transparent to the light but intercept a portion of the incoming rainfall. So the rainfall is much lower under this plot, this rainout shelter, than outside. And we compare plant biomass and plant species composition in this street plot with the control situation outside the rainout shelter. Las Chilcas is one of the experimental sites of the nutrient network, which is a coordinated experimental network established in different grasslands across the globe. In these experimental sites, we have the same experiments, so using the same protocol, we are studying the impact of nutrients and herbivore exclusion. As a control of plant biomass, there are bottom-up effects or controls that are the resources for the plants, and top-down controls that are related with the herbivores. Here we establish a combination of nitrogen, phosphorus, and potassium alone and combined to know the co-limitation of plant biomass production. So in our experimental plots, we measure every year the plant biomass and the abundance of each species, plant species, to estimate diversity index like Richner's, Simpson, Shannon, and this is a way that we, plant ecologists, use to have an estimation of the diversity of the plant community. With all this information that we are collecting by our field experiments, we are enlarging our understanding of how grasslands are functioning, the main effect of global change driver, and the manage that we can implement to increase forage production, which is very important for the livestock production in the Pampas grasslands. If you want to find out more information or research questions, you can visit our webpage of the Institute of Plant Physiology and Ecology at the Faculty of Agronomy at the University of Buenos Aires. Thank you very much for your attention.
Fantastic. So much fun to see these sites. It's now time for the panel discussion and question and answer session. Please post your questions for the researchers using the Q&A button rather than the chat. Our college classes have a time constraint of 50 minutes, so we're initially going to give priority to their questions. After that, we'll answer questions from other attendees until the top of the hour. As a reminder, students, please type the word student before your question and make sure that your full name is visible. The panelists are going to try to keep their answers to a minute or less so we can get to as many of these great questions as possible.
To get us all started, I'd like to hear from all of the panelists briefly on the same question. Let's start with Cindy Brown, then Scott Collins, and end with Laura Shadian. Remind us, what drew you to grassland nutrient research in the first place?
Hi, thanks, Carrie. I think what really drew me to this was the opportunity to learn about the effect that some of human activities are having on these on this system that I love so much. So that was probably the first driver, and then also how that will can inform our management of these systems to minimize those impacts.
was also the opportunity to work with a really great group of scientists. That was a real draw for me as well. So all those things together made it irresistible to do this work in the shortgrass steppe.
Hi, so when I started off in ecology, I assumed I would work in forests because I grew up in the eastern United States. I went to school in Ohio, and I got my PhD in Oklahoma, where there are a lot of forests in the eastern part of the state. And so what happened was I realized that one, grasslands were being neglected in a lot of ecological studies. And secondly, you can do a lot of cool experiments in grasslands, and you can learn a lot from those systems in a relatively quick amount of time, and they're high in diversity. So they're really cool experimental systems to begin with. And so that's why I started to work in grasslands. And then it's well known that certain nutrients are limiting to a lot of organisms, especially terrestrial organisms, and nitrogen is one of those most limiting nutrients. And so what happens when all of a sudden there is an abundance of the most limiting nutrient? It just becomes a very interesting question. And how does that factor change as you go across ecosystems on a global basis? It makes for a nice, powerful story.
In my case, I also love grasslands and open areas in general, so the grass shrub steppes too. And what drew me into this global coordinate network was the possibility to compare the effect that I used to observe and to measure in my grassland, in my country, with what happened in different grasslands across the globe. So this was a great opportunity to generalize the effect and the patterns that we see and we ecology measure, and to find out which are the main drivers of the response to plant community, plant diversity, and plant biomass to nutrients and herbivores and the other global change drivers that we use to include.
Fantastic. Thanks for those insights. I'd like to follow Carrie's question with another for each of you to answer. So during the time that you've worked at your research site, have you observed impacts of climate change? And if so, what are a few observations you've made about how your grassland site is changing in response to changes in climate? Let's see, let's start with Scott, and then Laura, and then Cindy.
Yeah, sure. So as I said in the video, the Seviata lies at an ecotone between two major vegetation types, the Chihuahuan Desert and the Great Plains. And so because we're at an ecotone, we're in a system that is going to be particularly sensitive to climate change because we're at the boundaries of species ranges. And what we have observed through very long-term, just simple measurements is that the Chihuahuan Desert grass black grama has increased a lot over the last 30 years. And the Great Plains grass, the one to the north, is not increasing. And so we're seeing a gradual movement, essentially, of Chihuahuan Desert species northward, which is exactly what you would predict in a region like ours, where it's getting warmer and drier. And so we have been able to relate a lot of these dynamics to changes not only in precipitation, but in what we call aridity, the dryness of these ecosystems, and the ability of air to sort of pull water out of the system.
What I identify in the Pampas, especially in the flooding Pampas where Las Chilcas is located, it's a higher frequency and intensity of flood. It's hard to say that it's just a consequence of climate change. I think it's a combination of climate change with land use change, because we have transformed the vegetation cover that change the amount of water that evaporates every year. So we don't know if it's just a consequence of climate change, but the frequency and intensity of flooding is increasing in our grassland.
So for us in the shortgrass steppe, which is part of the Great Plains, north of where Scott is in New Mexico, we aren't really measuring changes in the natural grassland, but there have been a number of studies that have been done to look at the response to elevated CO2 and warming, actually in another part of the Great Plains grassland, which is the mixed grass prairie. I think I would go along with what Laura was saying too, in that there is an interaction between land use change and climate change going on. So what we saw in those studies and what we're seeing in places where they remove the herbivores is a shift from a system dominated by the warm season grasses to one dominated by the cool season grasses. So that's a pretty major change in our system, and that's in terms of the climate change that we might be able to measure. There's some indication, as predicted, that we are getting larger, less frequent precipitation events, which actually means there's more effective rainfall, meaning effective. We say that rainfall events that are greater than about 10 millimeters are the only ones that actually become available for the plants to use. So that's effective. So those are some of the changes we're seeing, in the background of having longer, warmer growing seasons and higher carbon dioxide concentrations. So.
Cindy Brown, that is, it basically answers this question, but I'm going to ask in case you have something to add. Somebody writes, are you to control for phenological impacts? For instance, how the increased growing season due to climate change and how it interacts with nutrient load increases?
That is a great question. And there are folks working in the nutrient network who are monitoring the phenological changes in their sites. That's not part of the core study. So I think we might not pick that up in the standard sampling until you start to get some changes in actually species composition as a result of the conditions, environmental conditions changing to favor some species more than other species. I don't know, Scott and Laura, do you have other things to add?
I've got questions to ask. I want to keep moving them along. So this is really fun. Scott, actually, I'm going to direct the next two at you. There are two questions about nitrogen. One is, it's really about, the relative rate of background nitrogen deposition relative to the treatments. And the other one is, given how dry your site is there foliar absorption because of N deposition? And with the water limitation, how limiting is that nitrogen in your system?
Yeah, those are very interesting questions. I actually don't know if these dominant species are using foliar nitrogen. It's certainly possible. The background deposition rates, although I was sort of pointing out the pollution effects that we actually witnessed down there in the refuge, the background rates are still relatively small compared to, say, what's happening in the Midwestern United States or around urban areas. And so the amount of nitrogen that we're adding in this experiment is enormous compared to background deposition rates. We had, I guess nobody ever mentioned it in the presentations, but it's 10 grams per meter square for those of you who are keeping score. And we're getting nitrogen deposition rates of around 0.2 to 0.3 grams per meter square from atmospheric deposition. But still the soils that is still a significant increase in nitrogen in that system. And so what was the other part of your question?
Sorry, I had to find my mic there. It was about foliar absorption because, foliar absorption and water limitations. So.
Water limitations, yeah. So there's a debate in a lot of grassland ecology sessions about, whether nitrogen is limiting or water is limiting or they're co-limiting. And I am arguing here that nitrogen is important and it does limit this ecosystem, but you have to have sufficient moisture in the system first before nitrogen limitation can become evident. So in years in which we have above average precipitation, we see a large increase in production on those plots where we add the fertilizer. So it's sort of a sequential relationship between the system, but you have to have water first before the nitrogen response can become evident.
Great. Now this one is directed to Lara Chatian. This student wants to know about plants in Argentina, grasslands, and if they're native.
Most of them are native, but we also have some exotic species that are invading our grasslands, and surprisingly, they are more common or more abundant in our nutrient addition plots. So they became more dominant with the nutrient addition, which is something that we are interested in study more.
Super. There's a student that asks about evolutionary history, and I want to direct this actually to both Laura and Cindy. You have very different evolutionary history of grazing at your sites, but you both have cattle now. Do they have different effects now because of the different historical grazers?
Okay, so at our site, I think the cattle are mimicking the effects that the bison would have had for the most part, maybe not down to the very detail, but the biggest, in our system, the biggest disturbance is actually removing the herbivores, removing these big grazing animals. So we do see shifts in species composition in the plants simply by removing the grazers. So the cattle are actually doing a good job of replacing the bison kind of functionally in the system at this point.
We had big herbivores in the Pampas, but they became extinct a long time ago. So now the big mammals herbivores are mostly represented by these exotic species like horses, cows, and sheep. And so they are playing an important role in our grasslands. And we know that they are important to determine the plant diversity and plant biomass.
I'll direct this question at Scott Collins, but anyone else who'd like to step in, feel free quickly. What has been the most surprising or unexpected finding you've observed from the NutNet plots at your specific site?
Well, I think the most surprising thing is that the dominant grass, we assume that all these species are going to respond positively to nutrient addition. And to find that one of the species that is increasing in abundance out there hates nitrogen deposition, and it actually can even decline on nitrogen fertilizer plots, sort of it allows us to ask a whole lot of other questions. So why is that happening? Is it something that it creates a soil reaction? Is it because the nitrogen deposition alters the microbial communities and the interactions with this grass? It's sort of typical when you set out with an experiment that you have a simple question: what happens if we add nitrogen? And you end up saying, well, this happens, and now there's 10 more new questions. And so we are doing a lot of work in this system to try to understand the role of microbial plant interactions and how those are affected by a number of factors, including nitrogen deposition, drought, and altered rainfall patterns, etc.
Something similar happened in the Pampas because we add a lot of nutrients, nitrogen, phosphorus. We know that plants love those nutrients. However, we found a very little effect on plant biomass, especially when we exclude herbivores. That's because apparently we have a light limitation with nutrient addition. So the response of plants, even in our very messy, more humid grassland, that the one that Scott is studying, or Cine, we saw very little effect of nutrient addition on plant biomass, and that surprised us a lot.
Yeah, and for us, I think just this shift to the loss of the dominant species, just like Scott was saying, that's pretty much of a surprise. And then also the increase of an invasive species, annual grass, that's never been a problem in this system in the past. And in our plots, it actually is spreading. So, yeah.
Super. So for students that have to leave now, thank you for joining us. We'll be emailing follow-up information. For other attendees, the panel will stay on through the top of the hour and continue to answer your questions. I'm going to direct the next one actually back to you, Scott, because I know that fire is really important at your site in particular. Does the fire frequency and intensity seem to be changing as a function of changes in grasslands due to climate change or possibly due to invasions?
The fire frequency in our system? No, I don't think fire frequency is increasing. But perhaps when fires occur, they're more intense now because of the climate conditions that they're occurring under. But our grassland system does not respond well to fire. It turns out that black grama, if you leave it alone and just let it sort of grow out there through good years and bad years, that black grama will increase quite a lot in abundance in these ecosystems. However, if you burn this system, it takes the black grama plants about 9 to 10 years to recover, to return to the same amount of biomass that they were producing prior to the fire. So it's not a very fire tolerant species, but it's very flammable in this system. We don't have native or ungulate grazers in the system. We don't have cattle out there. So the grazing pressure is relatively low. So there's a lot of fuel that accumulates in this system. And so fires are common. We have lightning caused wildfires that do occur in the system, but they're, the fire frequency is around every 10 years or so, and we don't think that's changing.
Laura Shadian, this person asks, do you have anyone looking into the mycorrhizal communities in these habitats? Given their importance to a lot of grassland species and their sensitivity to nutrient deposition and changing climate, it would be interesting to see if the mycorrhizal communities are shifting in any way in their species composition or function.
That's a great question indeed. Yes, thank you for asking. We are looking at the mycorrhiza indeed in our nutrient combination plot. And I don't have the whole results already to tell you, but the preliminary results looks like the mycorrhiza are less abundant when we add phosphorus, which we think that makes sense because that was what we expected, because the symbiosis with mycorrhiza has a cost for the plants. And it makes sense that when we add phosphorus, the plants are less prone to have this symbiosis with mycorrhiza.
Super, thank you. Cindy, do you see a large buildup of thatch in ungrazed treatments? And is it a major pathway to decrease species diversity?
Yeah, that's a good question. And we do, especially though, in the fertilized plots. So there's an important interaction there. It's not so much of an issue when the plots are not fertilized, but when they are fertilized and there's a lot of biomass production, especially in wetter years. And then you get this thatch buildup the next year, which leads to reduced germination and growth of the plants after that. So yeah, we do see that.
Laura Shadian, I have another question for you. This person is asking about below ground production. Are you collecting data on below ground net primary production or below ground biomass at your NutNet sites?
Yes, we collect data on below ground biomass, which is not the same as below ground primary production, but it's still a proxy in our experimental plot. But we didn't find an effect of nutrient addition on below ground production. What we did find is that below ground biomass in the roots, but also the carbon in the organic matter increase with our herbivores. So when we exclude the cows, the amount of carbon below ground decreases, at least in the first centimeter of the soil.
There are two questions here that are really about generality. One of them is, are there responses to fertilization in grasslands around the world similar? And the other is asking about how we can use distributed networks to answer general questions about ecology. Scott, do you think you could tackle those?
So are there generalities that come out of these things? You'd be better to answer that, Elizabeth, yourself, actually, as one of the head honchos of the nutrient network. But I think there are generalities that come out of these systems. It's one of the cool things that we see when we do these cross-site experiments is the variety of systems that people actually work in. Many of them are quite variable in the kinds of grazing history they've had or constant grazing that's going on in those systems. But from that, we can see that there's a certain amount of, in general, increase in biomass on average across all these sites and a decrease in diversity when we add nutrients to these systems. So there is that kind of very general and broad pattern. What becomes interesting is actually looking at the sites that don't fit that general broad pattern, and then we can ask questions about why are these sites responding differently than what we would anticipate and what is sort of like the global average in that case. As far as these global experiments, I think they really do extend the realm of inference in what we can learn about nutrient limitations. There's another network called NutNet in which we're all doing drought experiments, and we're learning there about how ecosystems vary in their response to drought, and then it allows us to ask new questions about why those systems vary. Why are there so extreme differences? And some systems are much more sensitive to drought than other systems. So I think there's a lot of generality that comes out of working in these networks versus just, I could come out to my site at Seviata and say, nitrogen has nothing to do with grasslands and grassland productivity. And then Laura would write a paper telling me I was wrong. And so we try to avoid doing things like that and trying to collaborate and coming up with more general patterns.
Super. I think you captured a lot of what I would have said anyway. So thanks, Scott. I think the ability to answer questions about generality, but then to really capture the variation among sites and try to understand why we get different answers at different sites. So I want to thank all of our speakers, Sini Brown, Scott Collins, and Laura Shetian. Thank you to the Organization of Biological Field Stations, to the National Science Foundation for funding this project, to all the wonderful people behind the scenes who made this possible. We'll be emailing all of you in the next few days with follow-up answers to any questions we didn't address today, a link to a recording of this event, a link to provide feedback and ideas for future events, what you enjoyed, other event themes you might be interested in seeing in the future, and suggestions for how to improve. Please share the virtualfield.org and our upcoming events with fellow instructors, students, friends, and colleagues. Past event recordings are accessible on Live from the Field page under virtual visits, where you can also find links to the websites of each site featured today and event resources, including publications from each researcher and other educational tools. Thank you and goodbye from the field. Panelists, can you please wave goodbye?
Thank you very much for all the students and participants for the great questions. That was.
Thank you for listening.
Yes.
Thanks, everybody.
And thanks to all of you.
Thank you, Elizabeth.
Because we leave.